My name is Ivo Gut. I direct the Spanish National Genome Analysis Center in Barcelona. I've been doing this for 15 years. As the name of the center says, we do genome analysis. This is our business, and sort of cover a lot of different aspects of genome analysis, going all the way from clinical applications into even diagnostic tests, covering different pathologies. We cover cancer, rare diseases, immune-mediated diseases, work single-cell technologies, a lot of different things, but also, Earth biogenome related projects of, getting ecosystems sorted. That's what we do as well. So we cover many things, but it's all around genome analysis in one way or another, with a lot of computational know-how that is in our center, and obviously a lot of technical setup, sequencing instruments, latest generation, in all sorts of different flavors and shapes. So one of the main difficulties that we have in what we sort of discover in our daily work, and how that could be translated into benefit for a patient, is that there is a lot of information that we generate. So, for example, if we look at a cancer genome, we will identify a lot of changes, genomic changes, somatic mutations in the genomes of these patients. And what will happen is that the majority of them, we have no clue what they mean. So we don't know what they mean. Then sometimes we identify genes that we notice that within groups of patients, they are recurrently mutated, and then they turn into very important genes to study further. The problem with it is that when you find such a gene, you usually make a lot of noise in the scientific literature, and you say, look, this is really important. This is as important as putting butter on bread. But then you do that for the purpose of, or people do that for the purpose of publishing a paper with high impact. Afterwards, whether it actually translates into patient benefit is a different story. So you have a scientific finding, and obviously in every issue of Science or Nature there is always a whole big list of them. And then when you actually get to the translation of that to the benefit of the patient, it takes either extremely long until a finding is actually validated or replicated, and then actually makes its way into some translation, or it turns out that the finding is not quite as important as was claimed, and sort of never goes anywhere. So this is the problem, that not a lot that is actually discovered then makes its way through. And the other issue is that if you look at cancer genomics, you will have thousands of mutations, thousands of things in a genome of a patient. And then figuring out which ones are the important ones and which ones are the not quite so important ones is also very challenging. And the majority, we have no clue what they do functionally. We don't know how they relate to the story of this patient or of the tumor of the patient. And then we end up with sort of a lot of unknowns in our analysis. So what do you do with those? And that's obviously a huge problem. There's too much information, not too much information for me. It's interesting to have that information, but it's a lot of information, and for a clinician, not necessarily useful information, but information that's important to sort of systematically pick it up. And that's probably one of the things that you have to think about doing with the data. You have to accumulate the data, organize it in a way so that it's well structured, and then sort of pile it up in studies. Have large number of patients with similar presentations collected, and then looking at the similarities, and that is sort of like one of the action points that you have to do. You have to organize the data in the best possible way, so that you can then, once you have enough data together, you can sort of figure out whether something is statistically significant, and you can actually use that significant observation for the benefit of a translation. So this is one of the key things that you have to go forward with. There is obviously, and this is something that's largely not done, functional follow-ups. So once you have an observation and you trust that statistically this observation is valid, then you would want to follow it up functionally. And because you have so many observations that might have a functional effect, the difficulty is how do you systematically go after statistically valid observations. And this is something that is not very often touched. People or scientists will pick up salient observations and will then validate those, but the difficulty we now have, there's not very many of them that are followed up functionally. And the other side of that is that we don't have a systematic way of doing functional follow-up. And obviously, if you could set up systems to do functional follow-up in a very systematic way, then you can make headway there. So this is definitely a second thing that needs doing when you are looking at the translation of an observation into a valid diagnostic test or a relationship to a selected treatment that you would want to apply.